Welcome to AP Biology. This video is on microevolution, the evolution of populations. Individuals cannot evolve. You're born with the genetics that you have. Populations evolve. Generation after generation, a population is going to change in order to become better adapted to the environment that it inhabits. The field of population genetics combines evolution with genetics in order to quantify, in order to actually measure the evolution taking place within a population. The gene pool is all the genes and the alleles in a population. Remember, alleles are the alternate forms of a gene. Genetic variation is at the heart of Darwin's theory of natural selection. Genetic variation is the fuel for evolution within populations. Variation is crucial for natural selection. Without different forms within the population, one form could not be selected over another form. Variation is measured in a population through average heterozygosity, and that's the number of genes that are heterozygous in a population. This is used because the heterozygote contains both alleles for a particular gene. Natural selection is going to select for advantageous traits in a population, and these traits are controlled by genes. So really we could say that natural selection is going to select for advantageous genes or advantageous alleles within a population. Evolution is going to occur when a population's gene structure changes. The gene structure is the ratio of homozygous dominant to heterozygous to homozygous recessive. Or we could say the ratio of dominant alleles to the ratio of recessive alleles. We can measure the ratio of genotypes within a population, and we can measure the ratio of alleles within a population using the Hardy-Weinberg Theorem. The Hardy-Weinberg Theorem states that the genetic structure of a non-evolving population remains constant. A non-evolving population is in Hardy-Weinberg equilibrium, and the following equation can be used to measure the frequency of genotypes and alleles. So this equation here, which is the Hardy-Weinberg Theorem, is used when a population is not evolving, and it can be used to measure both the frequency of genotypes and the frequency of alleles. When a population is not evolving, we say that it is in Hardy-Weinberg equilibrium. The equation is p squared plus 2pq plus q squared equals 1, or it equals 100% of the genotypes or alleles in the population. In the equation, the p stands for the dominant allele, capital A here. The q stands for the recessive allele, little a here. So p plus q equals 1, because all the dominant alleles plus all the recessive alleles is going to equal 1. P squared is the frequency of the homozygous dominant. 2pq is the frequency of the heterozygous, and q squared is going to be the frequency of the homozygous recessive. We'll perform many Hardy-Weinberg problems. Whenever you perform a problem, you always begin with q squared, because we always know the frequency of recessive individuals. If a person expresses the recessive phenotype, they have to be homozygous recessive. So we automatically know q squared. Once you know q squared, you can take the square root to get q. Once you have q, you could determine p, because p plus q equals 1. Once you have both p and q, you can then fill in the rest of the Hardy-Weinberg equation. Now, the Hardy-Weinberg equation is only used when the population is not evolving. So why would you use it to measure evolution taking place within a population? Well, if you use the Hardy-Weinberg equation at time A, and you measure the frequency of alleles and genotypes. And then you use the Hardy-Weinberg equation at time B, and you measure again the ratio of genotypes and alleles, and those ratios change. We were then able to measure and quantify evolution taking place within that population. In order for a population to be in Hardy-Weinberg equilibrium, 5 conditions need to be met. They need to have a large population size. That population needs to be completely isolated from other populations. Mutations cannot take place. Mating needs to be completely random. And natural selection cannot take place. If these 5 conditions are met, a population will not evolve and will be in Hardy-Weinberg equilibrium. Evolution is a generation-to-generation change in a population's frequencies of alleles or genotype, a change in the population's genetic structure. Therefore, a break in any one of these 5 conditions will cause a population to leave equilibrium and will cause it to evolve. A population needs to have a large size in order to stay in equilibrium. Small populations go through genetic drift. Genetic drift is when random change has a large effect on a small population. We can see 3 circles, and this first circle here on the left has a large number of marbles inside of it. So if you change the color of 1 or 2 marbles, it's not going to have a large effect on the frequencies within that population. As that population becomes smaller and smaller, a change in just a few marbles is going to have a larger effect on the frequencies within that population. Genetic drift can be caused by bottlenecking, or it can be caused by founder effect. Bottlenecking is when an event drastically reduces population size, an event like a natural disaster. Here you see a bottle filled with 3 different colored marbles. You have blue marbles, white marbles, and yellow marbles. An event takes place drastically reducing the number of marbles in that population. You can see that this new population, which is much smaller, is not going to reflect the same frequencies as the original population. Due to bottlenecking, due to simply just a reduction in population size, we have a change in frequencies within that population. Microevolution took place. Founder effect is when a small subpopulation gets up and moves away from the original population. This small population is going to have a different ratio of genotypes and alleles than the original population. Since the genetic structure changed, microevolution took place. We said that a population needs to be isolated. The opposite of isolation is gene flow. Gene flow is when 2 different populations begin mating. So genes are flowing between the 2 populations. This is going to reduce the differences between those populations. Since the populations are still changing in genetic structure, microevolution is taking place. We said no mutations can take place. Mutations are an important part of evolution because they are the raw source by which new variation is created. Mutations are important for evolution, but mutations that are passed on do not happen very often. We said that mating needs to be completely random. The opposite of random mating is non-random mating, or what we call sexual selection, the differential mating of males and females. There's 2 types of sexual selection. There's intrasexual selection and there's intersexual selection. Intrasexual selection happens within a sex, and we usually call it male competition. Males compete for the right to mate with females. Therefore, males that are larger and stronger and overall have better genes are going to win the competition and win the right to mate. Intersexual selection happens between sexes. We call this female choice. Females choose which male they allow to mate with them. Females choose certain traits that are indicative of health and of having healthy genes. In nature, we usually see that the males are brighter colored and they are fancier looking. Females use this information to choose a mate. Both intrasexual and intersexual selection are going to create sexual dimorphism, the differences in appearance between males and females. Finally, we said that in order for a population to be in Hardy-Weinberg equilibrium, natural selection cannot take place. Natural selection is the major force causing populations to evolve. They evolve to become better adapted to their environment. An individual's fitness, or Darwinian fitness, can be measured. An individual's fitness is an indicator of how strong their genes are, and it's measured by the contribution an individual makes to the gene pool, or how many children they have. The individual who produces the most children is going to have the highest Darwinian fitness. They are going to be given a rating of 1. Everybody else is given a number from 0 to 1, depending upon how many offspring they produce relative to the most fit individual. So if I'm the most fit and I produce 10 offspring and you produce 5, you will be given a fitness of 0.5. I'll be given a fitness of 1. If you produce 3 children, you're given a fitness of 0.3. Natural selection can pull a population in different directions. There are 3 different modes of selection. The first mode of selection is stabilizing selection. In stabilizing selection, the average individual is favored, is selected for. We see stabilizing selection with the average birth weight. In human beings, the average birth weight is about 7 pounds and is the most advantageous birth weight. The next mode of selection is directional selection. In directional selection, one extreme is favored over the other. We saw directional selection take place with the evolution of a long neck in a giraffe. All the giraffes that had the longest necks were the giraffes that survived and reproduced. Generation after generation, the population of giraffes evolved to have longer necks. In disruptive selection, both extremes are favored. So you're going to see differences accumulate in a population, and this could eventually lead to the creation of 2 new species. Why is genetic variation so important for natural selection? Darwin said that variation is the fuel for natural selection. Without different forms, one form cannot be selected for over another. Quantitative characters are traits that have much genetic variation. They vary along a continuum. They are the opposite of discrete characters. Discrete characters exist in an either-or basis. Some discrete characters would be things like tall versus short, pink versus white. If you look at the trait of height in this flower that's growing along a mountain range, you can see that there is a lot of variation in flower height. This trait is a quantitative character. It's created through polygenic inheritance, and you can see that different heights are selected for at different elevations along the mountain range. How is genetic variation created? Genetic variation is created through 2 means: mutation and sexual recombination. Mutation is an important source of variation because it's the raw source of new variation. However, only mutations that take place in germline cells can be passed on. So mutations play a pretty negligible force in evolution. The only organisms that evolve quickly through mutation would be bacteria and viruses. Sexually reproducing species create genetic variation through sexual recombination. Genetic recombination takes place during meiosis, and it mixes up all the genes. The three forms of genetic recombination during meiosis are crossing over, independent assortment, and random fertilization. In a way, natural selection tries to extinguish one of the two alleles. One allele selected for over the other allele, so it's going to increase in frequency within the population. What stops the other allele from being wiped out? The fact that we are diploidy. We have two alleles for every gene. A heterozygote is going to contain both alleles, even though they only express one of them. Therefore, the heterozygote keeps both alleles within the population. Balanced polymorphism is the ability of natural selection to maintain diversity. Here we see three examples of balanced polymorphisms. One's called the heterozygote advantage. This is where the heterozygote is selected for over the homozygous dominant or homozygous recessive. We see this with the trait of sickle cell anemia and malaria. Individuals who are homozygous dominant will not have sickle cell anemia, but will be prone to malaria. Individuals that are homozygous recessive will not have malaria, but have sickle cell anemia. Individuals that are heterozygote, we refer to them as having sickle cell trait. They do not have sickle cell anemia, and they are also resistant to malaria. So in this case, the heterozygote will be selected for over the homozygous dominant or homozygous recessive. Hybrid vigor. Often when breeding plants, especially crop plants, we breed a homozygous dominant with a homozygous recessive, and we create hybrids. These hybrids have superior quality and help to maintain genetic variation. Finally, frequency-dependent selection is when what trait is successful depends on the number of individuals with that trait. An example of frequency-dependent selection could be seen in the anatomy of cichlid fishes. Here we see a population of cichlids, and there are two types. There's one type of cichlid that's going to guard its right side, and there's going to be a cichlid that guards its left side. Finally, our last example of balanced polymorphism is frequency-dependent selection. This is when the reproductive success declines when numbers increase. This is going to keep both forms of a trait within a population. We see this with the anatomy of cichlid fishes. There are two types of cichlids. There are right-mouthed cichlids and there are left-mouthed cichlids. This refers to the way that they latch on to their prey. They either sneak up on their prey and latch on to the right side, or they latch on to the left side. Well, if the left-sided cichlids are beneficial at the beginning and there are more of them in the population, eventually the prey fish will begin defending that side. When that happens, the cichlid fishes that are right-mouthed are then going to be selected for. So you can see this process that's going to keep going back and forth between the two types of cichlids is going to maintain both forms within the population. Finally, just some side notes about natural selection. It's important to remember that natural selection cannot fashion perfect organisms. Organisms are all locked into historical constraints. New traits and new alleles can't just arise on demand. That a population can only modify pre-existing traits. They are locked into their ancestry. Most adaptations are often compromises. Finally, not all evolution is adaptive. We know that genetic drift can have an effect on a gene pool just due to having a small population size. This concludes this video. Thank you.